Asia, and good evening to all of my friends here in the US and some people in between too. Welcome to this live broadcast of the last of the 4-part webinar series on next generation sequencing in clinical research. I am Krishnan Alampalam, and I will be your moderator for today's event. Before we begin, we have a couple of announcements. All products mentioned in this webinar are for research use only. These should not be used for diagnosis, prevention, or treatment of any disease. For additional information, please visit us at www.qiagen.com or talk to your tech support or your local distributor. To ensure that everybody can hear us clearly, we have muted all the lines. So to talk to us, you can do that only through the questions box that appear on the lower right-hand side of your monitor. So if you have any questions or you want to talk to me or the speaker, please enter it in the questions box. For now, just to make sure that you can hear me clearly, please enter yes or no in the questions box, so I know that you can hear me clearly. Okay, thank you. I can see some of them. So again, now you know where the questions box is, so feel free to use it anytime. This webcast is designed to be very interactive, and we want to encourage you to ask questions anytime during the event. As soon as the question comes to your mind, please enter it. That way, we can answer it in the order it will be received. We are very delighted to bring this educational webinar series on next generation sequencing in clinical research. Now, NGS is increasingly be used in clinical settings, and there are often questions of what to do, how to do, where to start, what are the regulations. So in this series, we wanted to bring experts in this field from various continents and share their experience of implementing NGS in clinical settings. As you see in your slide, in the first slide, you have Dr. Funk from Harvard Medical School, who has implemented next-generation sequencing in clinical research in the US. Same way, Dr. Pepper, who implemented next-generation sequencing in cell-free DNA from UC Davis. In part 2, we had Dr. Johan de Bono from Royal Marsden, Britain, who talked about translational research and prostate cancer. And today, we have Dr. Nishihara, who will be talking to us on the clinical sequencing system to classify meningioma. So let me introduce you to today's speaker. Dr. Hiroshi Nishihara received his degree in medicine and PhD from Hokkaido University School of Medicine, Japan. He did his postdoctoral work in molecular pharmacology at University of California, San Diego, under Dr. Paul Insel. After this fellowship, he returned to Japan and has been a faculty in the Department of Translational Pathology in Hokkaido University. He has authored and co-authored numerous publications in the field of oncology. Today, Dr. Nishihara will describe his experience of using targeted amplicon sequencing to develop a rapid clinical sequencing system to identify and confirm meningioma genotypes in just 7 days. In addition, we will also talk about details about the 3 meningioma categories that he has genotyped and the genes involved in this research. With that, I would like to turn it over to Dr. Nishihara for his presentation on developing a rapid clinical sequencing system to classify meningioma. Okay, Dr. Nishihara, please take over.
Okay, thank you. My name is Hiroshi Nishihara. I'm speaking from Sapporo, Japan now. So Today I'm going to introduce you our rapid clinical sequencing system to classify meningioma. And also I'd like to introduce you about kind of the biobank system which can promote this clinical sequencing system in our university hospital. Okay, first of all, I'd like to tell you about our university, and so I think you can see this the map of Japan, and this Sapporo is located in the North City in Japan, and then our university initially has been built by Dr. Clark as a Sapporo Agricultural College a long time ago. And then probably you know about the Sapporo as this beer. I think it's going to be very good beer, so you can enjoy the taste. Actually, anywhere in the United States, you can enjoy this beer. So also in our university campus, we have a very beautiful nature, and also you can see the very historical and very beautiful buildings. And also this is symbolic trees in almost middle of our campus. So actually, I'm now working in this very beautiful and sophisticated university. So next I'd like to say about myself. Actually, I'm a pathologist, and every day I'm doing making the pathological diagnosis, and sometimes I perform the autopsy. And also recently, I'm taking care of the biobank system in our university. And also, I just start the sequencing using the next-gen sequencer, and also now trying to establish the individualized medicine based on gene profile. So during this job, I established a good clinical sequencing system for, especially for the brain tumor. So I'm going to introduce you about that today. So first of all, I'd like to say a little bit about the background. So as you know, the development of the genomic medicine now promote the individualized medicine. Actually, we integrate the multiple patient genomic data, and also we should get high quality clinical information, and also we can perform the biostatistical analysis based on this profile. And for first part, actually, we can develop the clinical science as a clinical research. And after that, now the phase has been changed to the medicine. So I mean, we should perform the clinical sequencing for individualized medicine. So I'd like to show in this two part today. So for first part, actually, as a clinical research operation, we established this active biobank in our facility. So already we have a very good biobank system in University Hospital, and we perform the, of course, banking and also gene profiling or everything. And the basic system of our biobank, actually we called now clinical biobank, means the hospital-based biobank. And then the concept is, as you know, getting the sample is one problem for the good biobank. So our technician in our facility actually help to getting this sample and also taking care of the adequate sample processing in rapid. And then part of this sample can be analyzed for clinical sequence, and the result can be rapidly feedback to the physician and also patient. And the rest of the sample it could be stored and also linked with good clinical information. And these samples can be good tool for the specific clinical studies And also the part of the sample can be transferred to another university or institute. And then these samples can be analyzed for kind of the government-oriented clinical study or pharmaceutical-oriented drug screening system. So if we can get the good sample, these samples can be used for the clinical sequence and also clinical researches. So we already started this clinical biobank system in our hospital. So the basic concept is like this novel system of the biospecimen repository, which can promote various clinical studies and also clinical sequence. So actually, this our biobank is for use, but not for storage. Actually, this is a big point and an important point. And also, the tissue sampling is completely based on the specific protocol. So these samples, almost all these samples, gonna be used. So we never discard these samples. So almost all samples gonna be used, and even for the clinical sequencing. So already we established the clinical sequence system in our university hospital. So briefly, I'm going to show you the protocol. When getting this tissue sample, we usually fix with this packaging tissue system, which has been sold from the QIAGEN. Sometimes we use formalin fixed sample and also a frozen sample. After the validation of the specimen by pathologists, actually we need to confirm this sample is good for the clinical sequencing or some specific clinical researches. And after that, we got a paraffin section, and we can extract the DNA or RNA using QIAsymphony automated machine for DNA extraction. And we performed the quality check using some staff. And after that, we performed the targeted sequencing. Currently, we use MySeq from Illumina, but we hope to use GeneReader in near future. And then after that, getting the result of the sequencing, we performed the analysis using our original pipeline, which has been developed by the local company, AmeriF. And then, actually, this is the next step. We doing some team conference with physician or bioinformatician, and we get the final report for the gene profile, and the result has been explained to the patient for the outpatient. So already, actually for the clinical research, we have already started a couple studies. So this is one example, JUMP study. This is the target of this study is a lung cancer patient, and we gonna obtain a lot of lung cancer specimen from many facilities throughout in Japan. Actually, the tissue specimen has been fixed and sent it using this packaging tissue container. Actually, this is very good stuff because we can handle this specimen at the room temperature, so very easily can be sent. And also, we have already started this clinical study, HER2-CRAC test. In this study, the target patient has been the lung cancer patient who carried HER2, RB2 mutation. After the clinical sequencing for HER2, we performed the treatment the patient with Herceptin. So this is now after one year, we already checked more than 80 patients. So based on this background, actually we now newly established a good clinical sequencing system for brain tumor. And today I'm going to introduce about the meningioma. Already we are now starting to develop this clinical sequencing system for another type of the brain tumor, example, for example, glioma. But today, this meningioma, the system for meningioma has been already established, so I'm going to introduce about this. So I think almost of you know about the meningioma, but a little bit I'm going to talk about the background. Meningioma is arising from the meningocerebral cells, and also it's the most common primary brain tumor, and they account for 25% of all intracranial tumors. And then already we know the loss of NF2 has been one of the important alteration. But recently, some reports show the mutations in TRAF7, KL4, AKT1, and SMO, beside the loss of NF2. So, but actually, people still don't know about the genotype of these genes and also the relation to the clinical phenotype. So we established a clinical sequencing system for meningioma to determine the genotype as a routine laboratory examination in addition to the pathological diagnosis. So the protocol is almost same as I told you before. So getting the sample by PFPE and getting the DNA, extracting DNA using the QIAsymphony, and they perform the targeted amplicon sequencing, and the result has been obtained. Before showing the result, just a little bit, I'd like to show you the detailed protocol of this gene-lead DNA sequencing target panel. Actually, we for this meningioma clinical sequencing system, we employ this gene-lead targeted panel. So as you know, this gene-lead system is kind of a multiplex PCR enrichment system for gene in human genomes. And the average amplicon size is 150 base pair, and only we need just around 10 to 20 nanograms of DNA. And then the turnaround time is very short. Actually, the protocol said only later I'm going to show the detail, but for target enrichment only takes 3 hours, and throughout this library making, just only we can finish in the 2 or 3 days. So actually, this gene system already provides some targeted panel system showing here. For example, actually already we have employed a couple this panel, such as this clinical relevant tumor panel. I think this panel include 24 targeted gene exon, and also recently we start to use this comprehensive cancer panel, which cover the 160 cancer-associated gene, and then most of the genes has been related to the development of the cancer or even treatment for the cancer. And then also the analysis system is based on these databases like this COSMIC or CLINBR. So this CLINBR is related to the patient treatment record. And also they provide very good informatics system, Ingenuity. We can use this Ingenuity. But unfortunately, for the meningioma, actually our interested gene doesn't covered in these already established panel. So we have to make the gene panel using this gene-lead DNA custom panel. But it's very simple. Just we select the gene and give the name of the gene through the website, and then the QIAGEN make a panel through the website, and they gave us a report. And after that, we just simply click for order, and we can get the primer set after 3 weeks. And this time, we designed for NF2 and TRAF7, KRF4, AKT, SMO, and also a couple of related genes. And after getting this primer set, we performed a multiplex PCR with genomic DNA, and then performed the amplification. And then we can get DNA libraries. And for these throughout turnaround time is only around 4 days. So making libraries, we spent almost 2 days. And the analysis, so I mean the sequencing for 1 day, and the analysis need 1 day. So we can finish within the 4 days. Well, actually, we spent a little bit, a couple of days for making final reports. So now we said we can finish in 1 week, starting from DNA isolation to reporting. And I'm going to show a little bit detail the workflow for this NGS clinical sequencing system. We get the meningioma sample in frozen or even raw sample at the operation room. We fixed with this tissue using PAX gene. And then after that, we make PAX gene fixed formalin embedded blocks for all specimens. Because of making these blocks, we would like to confirm the histology. As you know, this confirmation of the tissue specimen is very important big problem. Sometimes we doesn't get the adequate part of the tumor, sometimes which contains only a necrotic part or even the normal part. If we use such the not good sample, the gene profile has been never used for the patient. So for every sample, we confirmed the histology. So confirming the histology, we need PFPE blocks. And after confirmation, we obtained the DNA from this PFPE blocks using a couple of paraffin slide. And after that, we making the library using the customized gene lead DNA panel, and we perform the NGS sequencing, and the result has been analyzed through gene variant coding service operated by QIAGEN through website. And also we use a Violet system, which has been developed by the company Amerief in Japan. So we can compare the result of these two analysis systems. So here's an example of the tissues. So as you can see, this is a fibrous type of the meningioma, or a little bit of papillary structure, or meningocele pattern of the tumor cells. We can see many types of the gene, many types of the tumor. And also we get these the genotypes like this NF2 loss or TRAF7 mutation, and also we for all specimen we got MRI images, and we can get clinical data for tumor location or enhancement of this tumor by gadolinium. And then the important analysis system for this meningioma sequencing is copy number alteration of the DNA, especially for NF2. As I told you, loss of NF2 is one of the significant gene alteration for meningioma. So we would like to identify the LOH, loss of heterozygosity, for NF2. So we use this gene-lead DNA-seq variant coding service for looking at this copy number alteration of NF2. Showing this one case of the result, this copy number analysis. This is target NF2. So this dot means that each lead for NF2 location. So in these cases, we can see the dot of each lead below the 2n baseline, and then the mean of the DNA level has been close to 1n. So the average is 1.2 to 1.4. So this means almost LOH of NF2, and then we also get the number of this score named as score P and score Q. And this score Q was used to set the threshold to identify the cluster of significant copy number changes. And then this system said over 50 of Q number is significant. But actually, we get more than 100 cases of meningioma, and we performed the copy number analysis for NF2 and obtaining like this. For example, this sample shows almost LOH of NF2, but I think this sample located in almost 2n baseline, so this means unchanged NF2 copy number. And then also we perform the fish analysis for NF2, and then we compare the result of this fish to the NGS score, Q score, and finally we obtained the conclusion: the Q score over 79 present the maximum Jordan index. So this Jordan index means that the maximum sensitivity and the specificity. For this analysis, we set the threshold Q score as 80. Over 80, we can say the copy number alteration has been identified. Based on this analysis, of course, we are looking at the mutations in these genes. We got the gene profile of 100 cases of meningioma like this. So based on this profile, we classified the meningioma into 3 subtypes. This is the NF2 type. In this NF2 type, this tumor has the copy number alteration, I mean the loss of heterozygosity of NF2 or NF2 mutations. Actually, important thing is NF2 mutation has been almost accompanied with NF2 LOH. And then next category is we call the Trackers type. In this type, we can only identify the mutations in this TRAF KL4 AKT SMO. So this group has no alteration of NF2 copy number alteration or no mutations. And the other group has no gene alteration in NF2 or even this Trackers gene. Currently, we cannot identify the driver genes for tumor genesis. So we just estimated to have some another gene alteration or even fusion genes in these groups. And then based on this profile, we categorize into three classes in NF2 type and the trackers type and no mutation type. And then important thing is if we look at the recurrence-free survival, so actually the meningioma patient has not so severe survivals. I mean, most of the patients can arrive longer period, but some of the patients suffered from the repeated recurrence. So we this time looking at the recurrence-free survival for these patients, and there is no recurrence in trackers type, but we identify the many recurrence in NF2 or NOC type. So this is very significant result for the neurosurgeon, because if the patient has been diagnosed as the meningioma in trackers type, the neurosurgeon doesn't worry about the recurrence. But if the patient has been diagnosed in NF2 type, the neurosurgeon have to watch closely the patient to find out the local recurrence. So the p-value is less than 0.05, so this is very significant. And also the tumor location has been associated with the NGS profile. And also tumor volume is actually this is related to this recurrence-free survival. The size of the tumor in NF2 type is much larger than the trackers type. So this gene profile is very important for the patient survival and also the decision for the treatment after surgery. So now we established this system. So after operation, we performed the routine pathological diagnosis, and also we performed the clinical sequencing analysis. And actually, even this clinical sequencing analysis has been finished in the 7 days or even 10 days. So not so taking long time, so we can give the result of this gene profile to the patient during their administration in a hospital. So I think this information is very useful for the post-operative management. So already in our university hospital, the neurosurgeons start taking these sample for clinical sequencing analysis, even in meningioma patient or glioma patient. For glioma, already we are now close to finalize the clinical sequencing system. For in near future, we can show the system for glioma also. And based on these results, in a university hospital, we are now we are going to start the new division for the cancer patient as this cancer genetic diagnosis division. So in this division, we take care of the outpatient for individualized medicine based on gene profile. Actually, in Japanese government insurance system doesn't cover the cost for such gene profiling, so patients have to pay by themselves for this gene profiling. But I think this system is very important for the cancer treatment. So actually, we are going to start this system within this year, and we performed in-house targeting system, and also we would like to, we should have this team conference for the final diagnosis with the doctor or technician, and also bioinformatician. And already the Department of Oncology and also Department of Cancer Chemotherapy Center start to collaborating with us. And after getting genotype, they gonna take up the patient for offering the clinical trial or even new drugs for patient treatment. And also for good data analysis and also annotation, we start a collaboration with a bioinformatics company, Mitsubishi Space Software, and they give us a very high quality analyze the data of the sequencing. And of course, we use a QIAGEN system and also a local company's system, so we can compare the result, and we also confirm the result with multiple analysis pipeline. And then we also start a clinical biobank study group in Japan. This study group taking care of SOPs for tissue handling and also SOP for sampling and also linkage to the clinical information, because this procedure is very important to establish the clinical sequencing. So we believe the good biobanking can promote a good clinical sequencing system. So we start the collaboration with these 4 universities, Hokkaido, Okayama, and Kyoto University, and Chiba University, and also QIAGEN supporting this study group activity. And then we start the networking of the clinical biobank in Japan. Just started in last year, and this year we can expand our activity to many other university, hospital, or cancer center, and we would like to establish this clinical biobank and also clinical sequencing system in Japan. Thank you for your attention.
Thank you very much for this wonderful, wonderful presentation, Dr. Nishihara. I appreciate that, and especially your insight on how the Japanese system, the medical system works. That's really very, very important to this audience, since this is a global series and people have an idea of how the Japanese system works. So I really appreciate that.
Thank you.
So I want to remind the audience that you should please go ahead and enter your questions as and when you have any at this point, so that we can answer it as and when we receive it. So let me start off with the first question we have here. Can you use FFPE specimen for your clinical sequence system?
Yes, I think this is one of the important points. Currently, we employ the packaging because the quality of DNA is extremely good compared to the FFPE. But we have to use FFPE specimen sometimes. And already we established the workflow using the FFPE, and we can say we can use FFPE now, but actually it's a little bit the quality of the data has been getting low, even in mutation profile, or actually the copy number analysis has been affected much more than the mutation. But we can get the good threshold and for the FFP specimen, so now we can use FFP also. But important thing is the quality of the FFP specimen. We cannot use sometimes the bad sample of FFPE, so we have to check the quality of the DNA obtaining from the FFPE before making the library. But now I can say we can use the FFPE also.
Okay, wonderful, thank you. No, I know that one of the questions that most often asked is that how do you validate this next generation sequencing results? What are the general strategies would you recommend, and what is your experience on validating results? And keep in mind, one of the things that I always get questions about is, yes, I like, should we use an orthogonal method like a PCR method, or should we use another instrument, another NGS instrument to confirm the results? What are your comments on that?
Yes, for the sequencing system, actually we now only use Illumina's MySeq, but I think we would like to compare the results using another type of the machine, so such like gene reader or even ion. We never compare with the result yet, but I think we should compare the result. And also, I have some colleagues who has a lot of the experience for the sequencing, and they said actually the result of sequencing is different from different by what kind of sequencer they use. So I cannot say which sequencer is good for this clinical sequencing system, but important thing is if we choose one sequencer, we should have the validated threshold or workflow for such sequencer. Then I think the important thing is the analysis of the sequencing data. So if we get the good analysis pipeline for the sequencing data, we can lead to the proper diagnosis of the genotype. For such purpose, actually, we employed at least 2 sequencing pipeline, including our original developed sequencing system, pipeline, and also using the Kaizen web system. And in addition, we are going to start to use the Mitsubishi Space Software's analysis pipeline. So currently we are going to compare 3 analysis pipeline, and we can compare that. We would like to obtain the much more proper result. So I think I would like to recommend people for clinical sequencing to use multiple clinical analysis system for DNA sequencing data.
Okay, thank you. What are the other target cancers in your for clinical sequencing?
Yes. Actually, in our pathology department, we obtained many types of brain tumor. So as I told a little bit in my talk, our next target is the glioma of the brain tumor. But also, we are now making the specific panel for the gastric cancer and also colon cancer, and also even pancreatic cancer. And of course, we performed the universal cancer panel, I mean clinical comprehensive cancer panel. So such cancer panel can cover for almost type of the genes, but one of the difference for this clinical sequencing is the targeting the fusion genes. We have to use RNA for detecting the fusion genes. Even using the RNA, it's not easy to identify the fusion genes. So I think for the sarcoma or some specific solid tumor harboring the fusion genes, we have to develop the good system for detecting such fusion genes. But we currently don't have the good tools for that. So actually already some big hospital, such as Memorial Sloan Kettering or even sequencing company, already provide the DNA sequencing system covering for the fusion genes, but they have to spend a large region for intron. So they have to perform using the HiSeq. But for clinical sequencing system in hospital base, I think we cannot have the HiSeq. So I would like to develop the small target panel which can also cover the fusion gene. Yeah.
Okay, thank you very much. That was the last question for today.
Okay.
And I want to thank Dr. Nishihara for giving this wonderful webinar and a very good insight on how it is done in the Asian, in Southeast Asia and Japan. So I really appreciate that. Thank you very much. And if there are some questions regarding the webinar slides and presentations, it will be sent to you within 48 hours to all the attendees. With that, I just want to thank everyone for being here today, and hope to see you in some other series very soon. Thank you and goodbye.
Thank you very much. まだリコーディングしてるのかな